Welcome to another episode from the Veterinary Research Channel. Today we delve into a fascinating study published in the Journal of Veterinary Internal Medicine in 2024, which investigated the intricate world of blood clotting, or hemostasis, in cats with hyperthyroidism compared to their healthy counterparts. Hyperthyroidism, a condition where the thyroid gland produces excessive hormones, is well known in humans to be linked with a state of increased blood clot formation and a higher risk of thromboembolism, a dangerous condition involving blood clots blocking blood vessels. For example, studies in people have shown that hyperthyroidism doubles the risk of developing blood clots in veins or arteries of the lungs and elevates the risk of ischemic stroke. This hypercoagulable, meaning prone to clotting, and hypofibrinolytic, meaning reduced ability to break down clots, state has been well documented in humans suffering from this hormonal imbalance. In the feline world, however, the connection between hyperthyroidism and thromboembolism hasn't been as clearly established. While two large studies looking back at cases of arterial thromboembolism in cats did find hyperthyroidism in a notable percentage of these cases, around 8 to 9%, it's not considered a widely recognized complication in cats. Previous research attempting to understand blood clotting in hyperthyroid cats using traditional methods has yielded mixed results. Some studies reported normal or even shortened prothrombin time and normal activated partial thromboplastin time, which are common measures of clotting function. Others found increased levels of fibrinogen, antithrombin, and von Willebrand factor, all proteins involved in hemostasis, in hyperthyroid cats compared to healthy ones. However, when researchers tried to define a hypercoagulable state based on having two or more abnormalities in these traditional tests, they didn't find a clear link with hyperthyroidism in cats. The authors of this study point out that these conventional blood tests have limitations. They don't fully account for the role of blood cells in the clotting process and often examine specific parts of hemostasis in isolation, rather than looking at it as a dynamic and complex process happening all together. These traditional tests might also be less sensitive in detecting a hypercoagulable state, and not as good at predicting how blood will actually clot compared to newer methods called viscoelastic assays. Viscoelastic assays such as thromboelastography, a point-of-care viscoelastic coagulation monitor known as VCM-VET, and rotational thromboelastometry, continuously monitor the entire process of blood clot formation, from the initial trigger to the final breakdown. They provide information about how quickly a clot starts to form, how fast it develops its strength, how strong it becomes, how stable it is, and how it eventually breaks down, giving a more comprehensive picture of what might be happening in the living body. Interestingly, a single prior study that used thromboelastography did not find significant differences in hemostasis between hyperthyroid and euthyroid cats before they received treatment with radioactive iodine. The role of platelets, small blood cells crucial for initiating and propagating blood clots, has also become a significant area of research in hemostasis. Two previous studies looked at platelet function in hyperthyroid cats. One of these studies, however, only looked at a small number of hyperthyroid cats, many of whom had other heart problems and were on medications that could affect platelet function, making the results difficult to interpret. The other study, using a platelet function analyzer, did not find any differences in platelet function between hyperthyroid and healthy cats, but it didn't thoroughly screen the cats for underlying heart disease, which could also influence platelet behavior. Given these gaps in our understanding, the researchers in this 2024 study set out to comprehensively evaluate both the overall blood clotting process and platelet function in cats with naturally occurring hyperthyroidism, compared to a control group of healthy cats of similar age. They used two different viscoelastic methods, the VCM-VET and rotational thromboelastometry, along with whole blood platelet impedance aggregometry, to test their hypothesis that hyperthyroid cats would show signs of altered hemostasis consistent with a potential state of increased blood clot formation. Now, let's move on to the findings of this study. The researchers enrolled 16 cats diagnosed with hyperthyroidism and 15 healthy cats over the age of 8 years. They carefully screened the cats to exclude those with other significant illnesses, or those receiving medications that could affect blood clotting. Blood samples were collected with minimal trauma under sedation, and various tests were performed immediately. The results from the point-of-care viscoelastic coagulation monitor, or VCM-VET, revealed some significant differences between the two groups. The hyperthyroid cats had significantly higher values for several key indicators of clot strength and firmness, specifically the clot amplitude at 10 minutes, the clot amplitude at 20 minutes, and the maximum clot firmness. This suggests that the clots formed in the hyperthyroid cats were stronger and developed more quickly compared to those in the healthy cats. However, no significant differences were observed in the clotting time, clot formation time, or the alpha angle, which are other measures of clot initiation and speed of formation, when using the VCM-VET. The rotational thromboelastometry, or ROTEM analysis, which uses 3 different activators to assess different pathways of coagulation, also showed notable differences. In the analysis using an activator called XSTEM, the hyperthyroid cats exhibited a significantly increased alpha angle, indicating faster clot formation, and a significantly higher maximum clot firmness, again suggesting stronger clots. They also had a significantly shorter clotting time, meaning their blood started to clot more quickly. The ROTEM analysis using another activator called INTEM revealed that hyperthyroid cats had significantly increased clot amplitude at 10 minutes, clot amplitude at 20 minutes, and maximum clot firmness compared to the healthy cats. These findings further support the idea of increased clot strength in the hyperthyroid group. Interestingly, the ROTEM analysis focusing on the contribution of fibrinogen, a key protein in clot formation, using an activator called FIPTM, showed even more pronounced differences. Hyperthyroid cats had significantly increased clot amplitude at 10 minutes, clot amplitude at 20 minutes, and maximum clot firmness in this assay as well. They also had a significantly shorter clotting time and a significantly decreased maximum lysis, which is a measure of how much the clot breaks down. This decrease in clot breakdown might also contribute to a hypercoagulable state. In contrast to the findings with the viscoelastic assays, the whole blood platelet impedance aggregometry, which measures how well platelets clump together in response to an activator, did not reveal any significant differences between the hyperthyroid and healthy cats in terms of maximal amplitude, an indicator of platelet aggregation. Now let's delve into the discussion of these results and what they might mean. The researchers concluded that their study provided evidence of altered hemostasis in hyperthyroid cats, as assessed by the 2 viscoelastic methods, characterized by increased clot amplitude, firmness, and faster coagulation times compared to the healthy control group. This pattern of findings, particularly the increased clot strength and potentially reduced clot breakdown, is consistent with a potential hypercoagulable state in these cats. The fact that 3 key measures of clot strength and firmness, maximum clot firmness, clot amplitude at 10 minutes, and clot amplitude at 20 minutes, were significantly higher in the hyperthyroid group, using the VCM-VET further supports this idea. Similarly, the ROTEM data showed a significantly increased maximum clot firmness in all 3 analyses, along with elevated clot amplitudes in the INTEM and FIPTM assays, again pointing towards a tendency for stronger clot formation in hyperthyroid cats. The study highlights that while both ROTEM and VCM-VET provide similar information about the viscoelastic properties of blood clots, they are not identical. The VCM-VET relies solely on contact activation to initiate clotting, whereas ROTEM and thromboelastography typically use different activating substances depending on the specific test. The authors noted that at the time of their study, there was no research showing whether these different viscoelastic assays could be used interchangeably in cats. Interestingly, the findings of this study, which suggest a hypercoagulable tendency in hyperthyroid cats based on viscoelastic assays, differ from the results of a previous study that used thromboelastography and did not find hyperthyroid cats to be significantly more hypercoagulable than healthy controls. While that earlier study did observe a trend towards higher maximal amplitude and overall clot strength in the hyperthyroid group, these differences were not statistically significant. The current study's authors point out that thromboelastography and VCM-VET are not directly comparable, as another recent study found that their results did not generally correlate well in cats. Furthermore, at the time of this research, it was unclear whether ROTEM results could be directly compared to those from thromboelastography or VCM-VET in cats. The researchers also considered the potential role of increased fibrinogen levels, which have been reported in hyperthyroid cats in other studies. Since fibrinogen is a major determinant of clot amplitude and strength, the increased values observed in both the VCM-VET and ROTEM data could potentially be explained by higher fibrinogen concentrations in the hyperthyroid cats. In humans, a strong link has been found between fibrinogen levels and specific FIPTM variables, such as clot amplitude at 10 minutes and maximum clot firmness, both of which were increased in the hyperthyroid cats in this study. The FIPTM assay in ROTEM is specifically designed to assess fibrinogen concentration and fibrin polymerization by inhibiting platelet function, so the increased clot firmness observed here strongly suggests a role for fibrinogen. Additionally, the finding of decreased maximum lysis in the FIPTM assay in hyperthyroid cats suggests a possible reduction in the breakdown of fibrin, the protein meshwork of a blood clot, but the authors caution that the significance of this isolated finding needs further investigation. The lack of significant differences in platelet aggregation between the groups, as measured by whole blood platelet impedance aggregometry, aligns with a recent study using a different platelet function test. However, the authors acknowledge several limitations regarding their platelet function analysis. They could only analyze samples from cats that had sufficient platelet counts on the day of testing, leading to the exclusion of some cats due to platelet clumping, a common occurrence in feline blood collected with the standard anticoagulant. While platelet clumping was observed in both groups, the degree of clumping was not quantified, which is a limitation. Another limitation was the use of a single concentration of collagen, the substance used to stimulate platelet aggregation, which might have influenced the results. The chosen concentration was intended to produce a submaximal response, allowing for the detection of differences between the groups based on pilot data. The study also had other limitations. The researchers' institution had not yet established its own reference intervals for ROTEM or VCM-VET, but the study focused on comparing the hyperthyroid group to an age-matched control group, rather than determining if the values fell outside a normal range. Furthermore, it's possible that the control group included cats with undiagnosed health issues, as they did not undergo the same comprehensive screening as the hyperthyroid group, particularly regarding blood pressure assessment. Systemic hypertension, or high blood pressure, is known to be associated with endothelial dysfunction, which can affect hemostasis and potentially increase the risk of thrombosis. In hindsight, the researchers also note that they did not measure prothrombin time, activated partial thromboplastin time, or the concentrations of fibrinogen and von Willebrand factor, which would have allowed for more direct comparisons with existing research. Finally, while the left atrium to aorta ratio, a measure used to screen for heart disease, was statistically different between the groups, no cat had a ratio above the exclusion threshold, making it less likely that subclinical heart disease significantly impacted the results. In conclusion, this study provides evidence of altered hemostasis in a group of hyperthyroid cats, characterized by increased clot amplitude, firmness, and faster clotting times as measured by viscoelastic assays. These findings suggest a potential for a hypercoagulable state in these animals. Interestingly, the altered hemostasis did not appear to be related to changes in platelet function in this study. The authors suggest that other factors, such as increased levels of fibrinogen or von Willebrand factor, which have been reported in previous studies of hyperthyroid cats, might be contributing to the observed differences in blood clotting. Further research is needed to fully understand the complex interplay between hyperthyroidism and hemostasis in our feline companions. Thank you for listening to the Veterinary Research Channel. A link for the reference paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.